of roughly 3 billion of those letters. It's elegant in its simplicity. The genetic code has 4 different chemicals. We substitute A, C, G, and T for those. We attach 4 different color dyes, 1 color for each of the letters of the genetic code. It's like just solving a jigsaw puzzle, only the jigsaw puzzle has, in our case, 27 million pieces. So it came in a very big box and there was no picture on the cover.
Putting the pieces of this puzzle together has provided knowledge that will enhance the quality of human life, and perhaps even extend it.
We're at the very fundamental first steps in a very powerful force. There's some relationship between aging and our genes. So if we can control those genes, will we be able to extend the aging process? Well, it's distant stuff, but probably. Knowing the human genome and mapping up how it changes is a major step forward in understanding the making of this wonderful human body that we've got. Through the history of time, the DNA sequence has been marching down through generation after generation of your relatives. And now it continues in your offspring.
Overnight, DNA from Inez's eggs and Daryl's sperm unite, and 13 of the 27 eggs show the telltale dimple that indicates success. It's working. With exquisite grace, 1 cell becomes 2, 2 become 4. Each duplicates the original, unique DNA. The enchanted progression of cell division continues. For 5 days, the embryos are monitored. Finally, the division creates masses of cells known as blastocysts, and any one of these blastocysts may become a part of the Pearson family.
This embryo here, if you look at the outer shell on this center embryo, it's very thin. The embryo is getting ready to hatch out of its shell. It's a very nice blastocyst. The inner cell mass is going to become the embryo itself, what you normally think of when you think of a baby. Arms, head, legs, toes, fingers. And actually this inner cell mass is where you find the embryonic stem cells, which are very much in the center of the genetic revolution that's going on right now.
Embryonic stem cells stand in the vanguard of human life. These magical all-purpose cells will eventually transform into every cell type in the human body. This extraordinary potential of stem cells has made isolating them one of the holy grails of science, although a controversial one. Dr. John Gearhart was at the forefront of that achievement.
These cells have 2 properties. One is that if you keep them in the dish, under certain culture conditions, they will continue to form more cells like themselves. So you can grow a room full of these embryonic stem cells, and they are undifferentiated cells. They all look alike. If you take some of these cells, though, and you put them out in different kind of growth conditions, these cells are capable of forming all the cell types that are present in the human body. What we are looking at here are heart muscle cells that are beating as a tissue.
Once, these were stem cells with uncharted destinies. Dr. Gearhart has directed their development into heart cells, now able to beat in perfect synchrony.
It's always been the dream of humankind that someday we'd be able to replace tissues in the body that were either damaged or diseased or simply worn out. But we really never had the starting material to do this. Now we have in the laboratory in our dishes, growing nicely, virtually all the cell types that are present in the human body.
They make a you. A stem cell can make you. That's pretty powerful. And I can control this stem cell and understand everything about it. Now we're set up to answer one of the first and basic questions about how a human is made.
As we've hoped, we have very well formed and ready to transfer type of blastocysts.
After 6 days of growth in the lab, Inez's embryos are ready for implanting. She's shown what might turn out to be her first baby picture.
Now, with embryos of this quality, if I put in 3, the chance of multiple birth may be as high as 40 to 50%, usually twins. If that makes you overly nervous, then I would suggest putting in 2, which still gives you a very good rate, but less multiples. What do you think about all that?
I think we'll go with the 2.
Okay.
Yeah.
Okay, so we'll do 2.
At first, I was going to just do 2. So, well, if one doesn't make it, then one will live. And, like, you know, at the last minute, I was sitting there thinking about it. I said, well, that's not that much either. I mean, for the amount of things that we have gone through, to just do 1 was not a very smart idea to do. And then we started talking about it and said, wow, 2's not that great either.
Okay.
So maybe I'll do the 3.
Now you want to do...
Yeah, I'll do the 3.
Well, it's okay.
Yeah.
I'll put in that new order for you. Bob, she wants the 3 now.
The 3 embryos are put into a single catheter and guided onto the lining of Inez's uterus.
Our placement is very good.
Well, this is what you hope for when you start. I really think it's a 50-50 chance for her at this point. You know, whether or not she's going to get pregnant, we'll find out in about 10 days.
I didn't get like really excited because they said, you know, there is a chance that it might not work, might not be successful, and we don't want you to really get your hopes up too high, so we just kept it like that.
Science has done its best, but it will be a long 10 days for Inez. 27 eggs collected, 13 fertilized, 5 viable blastocysts. 3 now offering Inez the chance to be a mother.